Chairman. Good morning, everyone. I'm from Osaka, Japan, which is the, one of the largest city in Japan. 2 hours flight from here. This is Osaka here. And, we are here in Hokkaido, and there are about 9 million people in Osaka and only 1,800 cows in Osaka. And there are 700 small animal clinics and only one large animal clinic. So we are the last Jedi. And today I'd like to talk about SIDA treatment for abortion prevention in some cases. Ultrasonography has allowed us to find out there are pregnancies with higher abortion rate. In this session, I'd like to talk about these 2 types, non-corpus luteum side pregnancy, NCLP, which includes transuterine migration, TUM, which is abortion rate 70%. And fetal membrane detachment, MD, which is abortion rate about 55%. Left picture is normal single pregnancy, and right is a non-corpus luteum side pregnancy. Please note positional relationship between embryo and corpus luteum. Transuterine migration means that ovulated this side, and at the end of the long way travel, implanted the other side of the uterine horn. In the case NCLP occurred in inseminated cows, it is considered that this TUM occurred. TUM usually occurs in multifetal animals. There is a role to equalize the number of the pregnancies on the left and right horn, but it is rare in cattle. The incidence of NCLP in inseminated cows is 0.4%, very rare, and 5 to 10% on embryo transfer. This number on ET varies greatly depending on the technician who is implementing it. In other words, this number is inferred to include cases of not only TUM, but also the transplant position is shallow and TUM easily occurred, and simply incorrectly transferred to the other side horn. Abortion rates were both around 70%, very high. The reason for the higher abortion rate of the contralateral side is that the local P4 concentration of the endometrium is lower than the corpus luteum side. P4 suppresses the motion of the uterine smooth muscle, promotes the development of the uterine gland, secretes uterine milk, suppresses immunity to the embryo in uterus, and produce a uterine environment suitable for the embryonic development. Next, fetal membrane detachment, MD. We can see MD when the fetus is dead. With no heartbeat, fetal melting and debris. In rare cases, however, an MD image may be observed, even though the fetus is alive. In these cases, we call on producers to pay attention to abortion, and more than half of them will abort. And this is also rare. Incidence is 1.5% of all pregnancies. I will deal with cases MD with fetus is alive in this session. The fetal membrane is immunologically non-self to the mother. During pregnancy, the fetal membrane is subjected to immune tolerance by the action of P4. In pregnant women, cells that induce immune tolerance called regulatory T cells, Treg, are elevated by P4. However, in abortion and premature birth, P4 decreases at the fetal-maternal interface, Treg decrease, and Th17 cells increase that induce the inflammatory response in humans. Supplementing P4 may induce immune tolerance and suppress inflammation, we hypothesized. We know SIDA increases the blood P4, so it may also increase the local P4 concentration of the non-CL side endometrium. Therefore, the purpose of this study is to investigate whether SIDA insertion reduces abortion rates at NCLP and MD. In January 2010 to October 2017, 117 cows in 16 dairy farms on which reproductive checkup is conducted by our clinic every month. 15 cows out of 56 cows with NCLP, 30 cows out of 61 MD, total 45 cows received SIDA for 30 to 40 days. Abortion rates were compared to untreated cows by Fisher's exact test. This slide explains how I decided the treatment period. The peak of the fetal death frequency is from 30 to 70 days after insemination in our past research. I thought that the treatment period should be adjusted to this. Actually, we started first early preg check from 28 days and second recheck with doubles as a sex check from 55 days. When I found an abnormality in the first preg check from 28 days, insert SIDA immediately and remove it in the second preg check, I thought that optimal treatment would be possible without putting extra labor cost. SIDA was inserted at the time of the first preg check, 28 to 42 days after insemination, and was removed at the second preg check, 55 to 85 days, so the treatment period was set to 30 to 40 days. Results. In the cases of NCLP, 73.2% is aborted if nothing was done, but it decreased to 40% by SIDA. And there was a statistically different, statistically significant difference. In case of MD, 54.8% had become 33.3%. But there was no statistical difference between them, only had a slight tended. All cows did not aborted delivered normal birth after completing the gestation period. We suggest that SIDA treatment may reduce abortion rates in dairy cows with NCLP or MD. In the case of NCLP, the external P4 by SIDA raised the P4 in the local tissue of the implantation area of the non-CL side uterine horn and contributed to making an intrauterine environment suitable for embryonic development. In the cases of MD, we suggest that exogenous P4 might maintain immune tolerance at the fetal membrane interface, fetal-maternal interface. That's all my presentation. Thank you.